So hello everyone, and welcome to the second lecture in the Applications of Artificial Intelligence in Medicine course presented by Ames. Today I am your presenter. My name is Chun. I'm the VP Education of Ames, and I'm sure everyone knows me by now because you've received a lot of emails from me. I am a third-year biochemistry student, so I actually know a little bit about drug design, and that is what I will be talking about today. So the outline of today's lecture will be, first of all, we will talk about property prediction in drug design. Secondly, we'll talk about de novo drug design, which is designing like an entirely new drug. And thirdly, we'll be talking about synthesis planning, as in how to make a new drug. First, we'll be talking about property prediction. So let's discuss what are the properties that we're actually looking for. So here I have listed some clinical candidate molecule criteria, which are the criteria that we look for when we're trying to see if a molecule is suitable to become a drug. First of all, we need to see if it is potent towards the particular biological target that it is specific for. As in, does it have the effect that we want it to have? And does it target the molecule or the protein or the enzyme that we want it to target? Secondly, we want to see if it is selective against undesired targets, meaning that it doesn't randomly interact with other targets in our human body and cause adverse reactions. For example, it could cause an immunological reaction where you have an allergy towards the drug, or it could affect a protein that we aren't supposed to affect, and that could cause another adverse sort of reaction. We want to avoid those things. Third, there are the ADMET properties, which is a collection of 5 properties, which are A, for absorption, as in how does the medicine go into our body and how is it absorbed into our bloodstream, for example. Secondly, it is distribution, and that is where the molecule will go within our body, as in like which organs, which tissues, and how will it permeate our body's tissues. Thirdly is metabolism, and that is how the medicine or the molecule is broken down within our metabolism. Like if we eat a pill, how does our digestive system process it, and how will it interact with the other organs in our body and change shape or be affected by different biochemical molecules. Fourth, we have excretion, which is how will the medicine leave the body. It is important because there can be side effects related to this, and we have to know this information in order to know how long the drug will last and how frequently you should take the drug, for example. And lastly, we have toxicity, which is quite self-explanatory. Obviously, we don't want a drug to be toxic, and we don't want it to cause any adverse effects more than is necessary. So we want to screen for toxicity when we're trying to find a new drug. So let's talk about the current process of drug discovery first. First of all, we use high throughput screening, which is a way of rapidly testing millions of samples for cellular or biochemical activity. And this is not an exaggeration. Like it is definitely on the scale of 10 to the power of 6 or 10 to the power of 7 reactions in a day that is being processed with these machines and detectors. So something that does have activity when you sense it is called a hit. And then if you have a hit, you must subsequently undergo more testing and optimization of the hit for certain objectives. For example, whether or not you can synthesize it easily, or the criteria that I mentioned in the previous slide, like for example, the toxicity and the potency and things like that. So we have to test for that. And lastly, it definitely involves a lot of automation right now, because as you can imagine, if we're testing millions of samples every day, that can't be done by humans alone. So it involves robots and highly sensitive detectors and automated data processing. And here in a picture on the right is kind of what it looks like. You have these pipettes and cells containing reagents, and then you can detect whether or not they react or not. So next, we will talk about the benefits of using machine learning for property prediction. So in machine learning, you have access to large data sets because the pharmaceutical industry generates large data sets during compound optimization when they're trying to find new drugs or produce a new drug for marketing. And this is good, but this can also be a bad thing because many members of the pharmaceutical industry see their information about drug properties as like a commercial asset. So they don't want to share it with other people because they don't want to lose their competitive edge. So this is a limitation because there's not a lot of well-labeled, good publicly available data sets that are free to use. So this is a limitation on the innovation of machine learning algorithms that can predict the properties of drugs. And secondly, you can also predict cross-target activities. So what this means is that if you have a drug that is already approved for one purpose, then you can use that other drug and see if it will affect a different target in the body. Like, for example, a different ligand or a different protein. And you can see if it affects it in the way that you want it to, and then this drug can be repurposed to do something else. One example is that there's many drugs that are being used to treat insulin that had originally different uses. And during the COVID-19 pandemic, there are some COVID-19 antivirals that are being researched because they are already approved for a different effect and a different purpose. And you can use this and see if it has any good effects on COVID-19. Of course, this is beneficial because then you won't have to go through a long and arduous approval process for a new drug because you already know that this drug has been approved and therefore it doesn't have like any adverse effects on the human body or that it isn't like really toxic or anything. So this can streamline the process of creating a new drug on the market. And this is important for situations like the COVID-19 pandemic, where we really need a rapid response time in order to decrease the potential losses and decrease like lives lost. So third, we can streamline the drug design process by saving money and time. As I mentioned previously, the current state of how drugs are designed is using high throughput screening. And this uses a lot of resources and it takes a lot of time. Like, as you can imagine, like you have to process many samples, millions of samples every day. And this takes a lot of resources and is very costly. So if you can use a machine learning and sort of visualize the properties of a drug that you're going to make, then you can screen for those drugs that are likely to be successful and thus decrease the expenses of actually testing it. So there's a high failure rate of drugs in clinical trials, and this process contributes to the price of drugs. And this is why many drugs are very expensive, because in research and development, many of these drugs take many years and a lot of money to produce. So this money has to be made back when the drug is sold, and that's why they're so expensive. If we can use machine learning to expedite this process, then it probably won't be as expensive, and we can give more people access to drugs that they need. And lastly, there are advances in processing power recently means there's increased efficacy. So a machine learning algorithm can process a large amount of data in a short amount of time. And this means that it's more efficient and also more accurate. Okay, so here are some currently available machine learning technologies that are being researched or that are being used for property prediction. So for compound optimization, as in like optimizing the properties of a certain compound in order to save cost or time or the ease of manufacturing it, there are support vector machines, which is visualized in the picture that I have on the right. And there are also random forests, which apparently, according to my research at least, it has the best performance. And there's also Bayesian learning. Next, for drug repurposing, there's the similarity ensemble approach, SEED, which basically it compares existing drugs to an assortment of drugs that already exist, and it sees whether or not they are similar enough to potentially be useful. This is a very gross oversimplification, but in my references section, you can read more on these if you decide that you find it interesting. And lastly, for property prediction, there are deep neural networks, and this has also been shown to be a very effective way of predicting properties, but I won't go into too much detail on the technical aspects of deep neural networks because that is covered in some of our other lectures in the curriculum. So you can find the recordings of those on our AIMS website. So yeah, going back to this image on the right here, this is a visualization of what a support vector machine does. Basically, it classifies something into one of two categories, and it does this by optimizing the properties of a particular item that is given, and it sees, does it fit into this category more or this category more? Now I'll go through what random forests are briefly, but I won't go into too much detail because, as I mentioned, there's like other lectures that talk about this more in our other AIMS curriculum lectures. So a random forest is basically a bunch of different decision trees. And decision trees, I'm sure many of you have seen or at least encountered them throughout your life before. Basically, you can see if something has a feature. For example, I have this highlighter. If this is a pink highlighter, then I categorize it as pink. And if it is yellow, then I categorize it into a different category. So since this is pink, it goes into that category. And then there are different branches that come off of that category. Like, for example, is it a light pink or a dark pink? And then so on and so forth. And then eventually you can categorize it very specifically. So in this way, you can find out whether or not a given item is like fitting into a category or not. So the decision trees themselves are an example of like machine learning, but random forests are a collection of decision trees. So these decision trees are generated by the machine learning model, and every decision tree may come to a different decision on whether or not this item fits into one category or another, or it will predict differently whether or not this item is a yes or no, for example. So the average of all of these predictions of all of the trees in the forest will be the average prediction, and that will give you the final prediction of the entire forest. And this is a more reliable way of classifying things than a normal regular decision tree. Yes. Okay, so one example of something that is example of machine learning that's being used in drug discovery is deep neural networks, or DNNs, for predicting toxicity. So as you can see here, here are some toxicophoric features that are identified from a data set by a neural network. And when it sees these like chemicals put together in a certain configuration, then it could be like, oh, I think these chemicals are going to be toxic because of these features. And toxicophoric features means that they are already associated with toxic properties. So it has been seen then that the neural network is a little bit reliable because once we see that it understands these as toxic properties, then we know that it is correctly identifying them because these are already known to be toxic properties by experts in the field. So here, let's talk about some barriers of using AI and property prediction. So as I mentioned before, the amount of data that you need for machine learning can be a good thing and a bad thing, because the more data that you have, the more accurate your prediction will be. But at the same time, the data has to be accurate and it has to be reliable and good quality data. Otherwise, your machine learning algorithm might end up being really bad. For example, it could be very specific to the particular data set that it's trained on, and it won't be applicable to other data sets that are slightly different, for example. So it won't be widely applicable. And many data sets, if they are not labeled well, as in they don't have the properties written on them clearly, then the machine learning algorithm won't know what they are and won't be able to understand it. So it's very labor intensive, obviously, to go through with a human expert and manually label these data sets. But if the data sets aren't well labeled, then the machine learning algorithm will be messed up. So one thing that you might want to consider is whether or not the data sets were made with the end goal of the AI in mind. For example, on the previous slide, we talked about toxicology. So if you have a data set that is labeled with the toxic properties of a molecule, then the machine learning algorithm that is trained on that data set will be able to predict well whether or not a molecule will be toxic because it is already labeled clearly in the data set. However, if you have any random generic, for example, database on chemical properties, then it might not be suitable for predicting toxic properties because it won't have the specific properties like labeled clearly. So a machine learning algorithm that's trained on it won't be really good at predicting whether or not a compound is toxic. And secondly, there are some predicted properties that are currently too generic to be used without the guidance of human experts. So as in our previous example about toxicology, some of these like structures, they're really generic and they show up a lot. So if they are not seen within the context of the larger molecule, and since the environment of the larger molecule and the overall shape affects whether or not they will be toxic, then these identifiers are a little bit too generic, and the deep neural network might have trouble actually classifying them correctly. So a human toxicology expert will be able to tell if these features are toxic based on the context that is presented in, like the overall molecule. But a deep neural network might not be sophisticated enough to tell yet. Okay, so now moving on to the next section, which is de novo drug design. This is, as I mentioned previously, this is making drugs that don't exist yet. So these are completely new molecules. And currently this is not widespread. And there's not a lot of machine algorithms that are currently being used to make new drugs. But the benefit of using it is that there's like an astronomical number of potential new drugs that we can discover. Like the number is truly difficult to conceptualize. It's like 10 to the power of 23 or something like that. And there's a benefit to using machine learning algorithms because, as I mentioned before, right now there's been advances in the processing power of computers. So they can process a lot of information at once, and thus they can explore the potential of a lot of these new drugs. So what I wanted to mention is that many of these machine learning algorithms will use the SMILES form of chemical structures. So I thought that was important for you guys to understand. What is a SMILES? What is the SMILES form? Basically, as you can see here, here's the structure of a drug, for example. And then with a bit of processing, you can name the drug in a specific way that it tells you the structure of it. So basically, as you can see here, I can't see very clearly because the zoom bar below is blocking my mouse, but I think you guys can see it clearly. So down here, you can write a string of ASCII symbols that represent the structure of the drug in a single line. And this is the way to tell the computer what the drug looks like. And that is how a computer understands the structure of a chemical. So yeah, this format is called SMILES. And then let's talk about current technologies in de novo drug design. Usually, here are some popular ones. There's a variational autoencoder, there's an adversarial autoencoder, there are recursive neural networks, and there's transfer learning. So these are all different approaches within machine learning that are being used for de novo drug design. But once again, I won't go into too much detail about them because that's not the purpose of today's lecture. So now we'll talk about some benefits and limitations to using machine learning and artificial intelligence for de novo drug design. First of all, the benefits. Of course, there's the speed of execution because designing new drugs is a very labor-intensive process, and comparatively, using a machine learning algorithm makes it much faster. And secondly, there is the ability to rapidly retrain or fine-tune the machine learning algorithm. If you have new experimental data that has come out, then you can rapidly retrain your algorithm, and then it can immediately be improved. So this can be used if you find that this algorithm is deficient in some area, and you can correct it that way. And next, there is scalability. So since there's a large processing power, as I mentioned, there's so many potential new drug structures that it's difficult to conceptualize and obviously it would be difficult for a human to explore all of them. So a machine can look through much more of those potential drug designs than a human can. And that way, then we can have more drugs coming out and do more research quicker and potentially like save more lives quicker, which is the ultimate goal, of course. And last, there is also the synthetic accessibility of the designs. What this means is that the designs have to be able to be actually produced through chemical methods. Like if you take one drug and then you react it with another, then it can change the structure of that drug, and then eventually it will give you the design that you really want. So how do you know if this drug is able to be made by chemical reactions or not? Well, if you have a machine learning algorithm, then the machine learning algorithm will take into consideration how the drug can be made. So the designs that it suggests will be actually able to be made chemically in like a manufacturing plant or something. So next, let's talk about limitations. So the new hypotheses for drugs that are created by a machine learning algorithm are not always based on known chemical pathways. So usually we design new drugs based on our pre-existing chemical knowledge of how drugs interact and what drugs already exist, and we think about those designs and then make new drugs that are similar to it or based on it. However, the machine learning algorithm might not do that. So it might use statistics and probability to create a new design that is completely novel and different, and something that like a human drug designer would not have thought of. So this is a limitation because there's less explainability in artificial intelligence drug discovery, because we don't know like why it decided that this drug would be good and whether or not their process has any flaws. So this is, of course, because it doesn't use the traditional pathway of discovering drugs. It's based on like statistics and probability, and only the machine learning algorithm really knows what's going on. So that is why it's a limitation, because we want to know if it's actually doing the correct thing or not. However, even though we don't know why it is producing these drugs that it is, it's still designing drugs that have desirable properties and do hold up. So they are like potentially life-saving, and they do appear to be accurate and good. So yeah, it's both, I guess, a limitation and a feature of it, I guess. So next, we will talk about synthesis planning. And what this is, as I mentioned before, how you will make a drug, basically. Drug synthesis is definitely something that is really important when you're trying to design a drug, because if you can't synthesize the drug, then you shouldn't be designing the drug in the first place. If you can't synthesize it, of course, it will never be manufactured and never go to market. So what's the point of it, right? So on the right here, I have a picture of what potentially a drug design might look like and what reactions. For example, you take this molecule and then you go here through one reaction, and then you go to this through another reaction, and then you go to this through another reaction. And eventually you can make all of these molecules that can be used as drugs. So there are three aspects of drug synthesis that a machine learning algorithm can help with. First of all, there's the prediction of the outcome of reaction. Like, for example, it can predict that, oh, if you react this molecule with some other molecule in certain conditions, it will produce this. And secondly, the prediction of a yield of a reaction, which is basically predict how much you can make. And this is important because of the cost of making a drug. Like if you need a lot of molecules in order to produce a drug, and if you need a lot of reactants that are very expensive, then it is less feasible. So lastly, there's retrosynthetic planning, which is basically when you work backwards from the drug that you want to make and think about what reactions will get it to that point. And then you work backwards and see what reactions will get you to that point and how you will do them and what reactants are involved, et cetera, et cetera. So here are some benefits of using artificial intelligence in synthesis planning. You can optimize for cost, which, as I mentioned, is a very important aspect, because research and design of research and development of drugs takes a lot of money, and that's why drugs are so expensive. Secondly, you can decrease the time spent on thinking about drug pathways, because I mean, if you've ever taken organic chemistry, you know how difficult it is to think about the pathways that you can synthesize a molecule from. And third, it's highly effective, and artificial intelligence has already thought of many different pathways that you can synthesize drugs from, and these pathways have been proven to be good and effective. So fourth, we can analyze large data sets. As I mentioned before, you can have a large library of potential drug reactions, and then an artificial intelligence can take into consideration all of that and come up with a good solution. And fourth, since artificial intelligence and computers now have such large processing power, you can explore many different synthetic routes. So you can optimize for cost, and you can see which route is the best, basically, for certain factors and objectives that you want to achieve. And next, it's actually simple, because simple for machine learning to think about drug design, because there is already algorithms that are very similar to it. And this is in chess and Go. So if you know, there's Deep Blue, I think, is the chess AI that has beat Garry Kasparov. And so there's already very advanced and effective machine learning models that can play chess and Go very well. And how this is similar to drug design and retrosynthesis is that you have to think about many steps and how it will get you to your eventual goal. And you won't be able to know really how this particular step brings you closer to your goal until you actually achieve it. So that is how it is in chess. And thus, it's also applicable to retrosynthesis of drugs. So since there's already machine learning algorithms that exist that are very effective, we can use them in drug synthesis as well. And lastly, you are able to prioritize templates. And what I mean by prioritizing templates is that if you already know that there is a certain way of synthesizing a drug that is already in use, and there's a certain chemical pathway that you can follow, then you can tell the machine learning algorithm to prioritize these templates, and it will be more likely to give you a pathway that is similar to it. And therefore, you can sort of sidestep the certain outcomes that are not as welcome or not as effective, for example. Oh yes, on the right here, we have a visual representation of how an artificial intelligence might go about making the drug from the start to the end. So as you can see here, the orange is the product, and it's the final product here. There's many different pathways here of how you can create this final product. And here are the different routes that it can take, labeled with intermediates and substrates. Yes, okay. Now let's talk about some of the limitations of using artificial intelligence in synthesis planning. So there's large differences between reaction classes, as in the different chemical reaction classes. And this is because the quality of your machine learning algorithm is dependent on the quality of training data. And as I mentioned before, well, there's not too many quality and training sets available out there. And often machine learning algorithms will use chemical public data sets such as Campbell. Yes, I don't know how to pronounce that really. And PubChem, which I'm sure everyone is familiar with. So these large publicly available data sets have a lot of information, as you can see in this diagram over here, takes information from all of these different sources. But the thing is that they might not all be labeled in the way that you want it to. So of course, as I mentioned before, this can cause many different problems in your machine learning algorithm. So For this reason, it might be better to use a combination of knowledge-based and machine learning approaches. And what knowledge-based approaches is if you have a library of pre-existing chemical structures and chemical reaction flows, then you can use this library and basically search through it like a search engine. And then you can find the optimal way of synthesizing your drug, for example. And if you combine this with machine learning, it might be a more reliable approach. And another limitation is that machine learning algorithms currently cannot develop novel chemical reactions. Like it can't think of new chemical reactions that have never been thought of before. So yes, and that is something that it needs more research. I think the goal of using machine learning in drug discovery is that you can have a tool that helps scientists discover new drugs. So ultimately, it is a collaboration between humans and machines. And this is the most effective and efficient way of doing things because you can ensure the quality of your results, and also you can make it more efficient and more effective. So that is the conclusion I want to end on today, and the end of my presentation. So any questions, please come at me.
I just wonder, will the new drug that designed by artificial intelligence, like would they have to go through the same test with the drug that by human design?
Pardon, could you speak more clearly and loudly? I couldn't really hear you.
Okay, so like will the drug designed by the machine have to go through the same like process as the drug that designed by the human?
Yes, so a drug designed by the machine will have to go through the same clinical trials and as a drug that is designed by a human. And it will have to go through a verification process just like every other drug, because the drug might not be as reliable or effective as we originally thought it would be. But through machine intelligence and artificial intelligence, we can find drugs that are more likely to have the properties that we want. So it's more like it makes the process more efficient because discovering the drugs is more efficient and less work, but you will still have to go through the same testing as drugs that are found by humans.
So like it just saves the time for like finding out the new drug, not testing it. Okay, I get it. Thank you.
I've also got a question.
Yes, please go ahead.
Okay. I was wondering if you know how prominent using ML to synthesize drugs is already in the industry. Like, is it sort of an experimental thing, or is it already being used quite a bit?
So actually, using computers to automate the process of retrosynthesis, as in finding chemical pathways, is already quite common, and there's a lot of these processes that also incorporate machine learning. But for de novo drug design, like designing new drugs, then it's less common. Right now, it's not very widespread. And predicting the properties of drugs, this is sort of more in the experimental stages, but it is a very common research topic right now. So it is my hope that in the future, it'll be much more common. Yeah.
Interesting, thank you.
Any more questions?